bago man lang tayo mag-ano sa RNA viruses natin, so let's just try to review the properties of our RNA viruses. So that when we are talking of the RNA viruses, now ito yung mga labile and general properties din ng RNA ha, kasi ang dami nating RNA viruses. They are labile and transient. And then most of these RNA viruses, they replicate in the cytoplasm. So take note that is not all, but most of them. The cells cannot replicate the RNA. Kaya dito nyo makikita yung positive sense and the negative sense, which was also already lectured to you by Sir Tom last time kung ano yung positive sense at saka negative sense. And these RNA viruses must encode and RNA, bakit hindi ko mabasa yung ano? Wala po yung RNA-dependent RNA polymerase. So if going back to your cytogenetics, di ba alam niyo yung mga enzymes that would be responsible doon sa ating multiplication or sa replication ng viruses. Okay? So the genome structure also of these RNA viruses determines the mechanism of transcription and replication. So they are prone to mutation, kaya nga, di ba, ayan yung kasama ang coronavirus natin, si SARS-CoV-2, dito sa mga RNA viruses. So you are all aware of the different strains now that we have with regards to the SARS-CoV-2 because these RNA viruses, as has been mentioned, prone sila to mutation. So RNA viruses except positive sense RNA genome, dapat may sarili silang polymerase. And yung mga negative sense RNA viruses, lahat sila ay envelope. Okay? Yun yung general properties ng ating RNA viruses. So we proceed with the first one, sabi nga ni, which is your picorna virus. Dito is overview lang ng RNA viruses because next meeting, ang ano na natin is we'll get into the different individual families of viruses. Yung next topic kasi should be on viral pathogenesis, but I will give you a recorded lecture. You can do an asynchronous learning for that one. So we proceed with the different DNA and RNA families. Okay. So ito overview lang ng RNA containing viruses, just like what Sir Tom has done last time dun sa ating DNA viruses. And first, of course, we have the picorna virus. So mapapansin nyo, lagi nandiyan yung size nila, yung symmetry, yung genome, and ano ba yung mga pathogens na kasali dyan sa mga families na yan. So picorna viruses, di ba sabi, they're small, although most of the viruses naman, di ba lagi natin sinasabi, ito yung maliliit na organism. So they're small, they're 28 to 30 nanometers, ether-resistant viruses. And symmetry, this cubic symmetry, following the different RNA viruses, we'll be discussing on the different virus structure. For the picorna, sila ay may cubic symmetry, and the genome is a single-stranded and positive sense. Yun nga sabi natin kanina, yung nagsiserve siya as your messenger RNA and is 7.2 to 8.4 kilobase in size. So, yung mga pathogens na under ng picorna viruses, we have our enteroviruses. Dito sa enteroviruses, dito kasama yung ating poliovirus, yung ating coxsackie, and ang ating ecovirus. So, ang mga enterovirus, they're acid stable and they have a lower density. Si rhinovirus naman, nandito yung ito yung nagpo-cause ng common cold. So, there are more than 100 serotypes sa rhinoviruses. So, they are acid labile in contrast to the enteroviruses. Sabi nga natin, they're acid stable, but they have a high density. So, for the picorna viruses infecting animals, include itong ating foot and mouth disease. Ito yung sa cattle. And ang encephalomyocarditis naman sa rodents. Okay, so overview lang yan ng picorna viruses. Later on, we'll get into the different viruses under picorna. So, for the Ba't ayaw mag-move? Ba't ayaw mag-move? Okay. Astroviruses. Next is the astroviruses. So, medyo kapareho lang din siya ng size ng picorna. Kung maalala nyo kanina, sabi niya, almost pareho nga. They're the same 28 to 30 nanometers. It's just that ang characteristic naman ni astrovirus is that meron siyang distinctive star-shaped outline sa kanilang surfaces. Parang yung nakikita nyo ngayon dyan sa picture. So, ang genome niya ay linear, positive sense, single-stranded RNA, 6 to 4, 6.4 to 7.4 kilobase in size. And ang human pathogen na kasama sa ang kasama sa astroviruses natin, we have those that cause gastroenteritis in humans and animals. Okay? So the next is our Khaleesi virus. So ano yung ating Khaleesi viruses naman? So
ang putang ina kasi may klase na ako ba?
Sorry. Okay, so for the Khaleesi viruses, again, the size, this is slightly larger to the picorna. Diba? Kanina sabi natin, ang ating picorna is 28 to 30 nanometers. So itong Khaleesi natin ngayon, slightly larger with a size of 27 to 40 nanometers. And ang kanyang characteristic naman sa kanyang surface is meron siyang cup-shaped depressions. So lahat ito makikita lang siyempre sa electron microscope. The genome is single-stranded. It's a positive sense RNA with 7.4 to 8.3 kilobase in size. Siya ay naked kasi wala siyang envelope. So, di ba sabi ni Sir Tom earlier, protein, which is your VPG. Kasi yan mga yan later on, then you will learn about the different proteins na nasa surface ng ating mga viruses. Okay. So you have there, not very common is your Mycoviro viruses. Their size small with icosahedral surface. Wala din siyang envelope, so the genome is linear, double-stranded, segmented, and totaling to 4 to 4.5 kilobase. And reoviruses, these are reoviruses of humans include rotaviruses, which have medium size, 60 to 80 nanometers. They're also ether-resistant, non-envelope viruses having icosahedral symmetry, and the particles have 2 to or 3 protein shells. So short spikes extend from the virion surface. The genome still is linear. It is double-stranded, segmented RNA, totaling to 16 to 27 kilobase pairs in size. So the replication occurs in the cytoplasm. So rotaviruses, which have also again, so manotice nyo na ngayon pa lang sa RNA viruses nyo, may mga characteristics sila, distinctive characteristic. So that with the rotaviruses, they have a distinctive wheel-shaped appearance and causes also gastroenteritis. And the cultivirus include also Colorado fever tick virus of humans. And next is you have your Arbor Viruses, which Sir Tom slightly also covered last time, di ba? Yung Arbor coming from arthropod-borne viruses. So that these viruses, now yung ating mga arthropod-borne, or this arboviruses and rodent-borne viruses, this is more of an ecologic grouping, not a virus family. of viruses. So the arboviruses, ito yung complex cycle involving arthropods, which will also be seen in succeeding slides, as vectors that transmit the viruses to vertebrate host. So ano yung mga arboviruses natin na nagkakos the human pathogens that belong to this group. So we have the most common, which is dengue. Then you also have the yellow fever, you have your West Nile fever, and encephalitis viruses. So rodent-borne viruses, they are transmitted in rodents without an arthropod virus. So ano yung mga na kasali dito? You have your hantavirus, infections, and Lassa fever. Sorry. So yung sabi natin, di ba, it's not really yung arboviruses, it's not really a virus family. So that we have here, under arboviruses, you have your arenavirus, virus, flavivirus, reovirus, rhabdovirus, and toga viruses. Now, yung mga toga viruses natin, they have a lipid-containing envelope and are ether-sensitive. Ang genome nila, they're single-stranded, positive sense RNA, 9.7 to 11.8 kilobase in size. Envelope variance measures 70 nanometers, so medyo mas malaki na siya. Particles mature by budding from host cell membrane. So examples of our toga viruses, we have here Eastern equine encephalitis virus, kasama si rubella virus dito, at ang ating alpha viruses. Actually, pag itong mga karamihan ng arboviruses natin na human pathogens, they're actually called your alpha viruses. Kaya yung classification. So, flavivirus, so they're an envelope virus cells also 40 to 60 nanometers in diameter. So the genome is single-stranded, positive sense RNA. Mature virions accumulate within cisternae of the endoplasmic reticulum, and most members are transmitted by blood-sucking arthropods. So examples natin dito, we have your yellow fever virus, you have the dengue viruses, and HCV. The arenavirus, these are pleomorphic envelope viruses, so their replication is in the cytoplasm with assembly via budding on the plasma membrane, and particles have a sandy appearance. Now, most members are unique to tropical America, and all human pathogens cause chronic infections in humans. Example ng arenaviruses, we have our Lassa fever. Now, coronaviruses, what is causing everything in our life today? These are envelope, envelope, and they're 120 to 160 nanometer particles. So that, di ba na-mention natin earlier, pag envelope, mas madali silang i-inhibit or patayin. Kaya nga, ang ating way of trying to inhibit the replication of these coronaviruses wherein your SARS-CoV-2 belongs is just by hygiene and alcohol. So the coronavirus genome is unsegmented genome of positive sense single-stranded RNA, 27 to 32 kilobase in size, and it has a helical nucleocapsid. It resembles orthomyxoviruses but have petal-shaped surface projections similar to a solar corona, kung saan niya nakuha yung kanyang pangalan na coronaviruses. So, the nucleocapsids develop in cytoplasm and mature by budding into cytoplasmic vesicles. So most human coronaviruses cause mild acute upper respiratory tract illnesses. So ano yung mga yan? We have the colds, tapos di ba maalala nyo, ang nauna dyan kasi before the SARS-CoV-2, we already had the SARS. Kaya nga SARS-CoV-2 ngayon kasi parang second time na ito. And then you have the MERS-CoV and yung ngayon na yung ating SARS-CoV-2. Now, kailan 2003 ang SARS, and then nung 2000, kailan ang ating MERS-CoV? 2009 yata, and then we have the SARS-CoV-2 now. Now, Enteroviruses also, which cause gastroenteritis. So makikita nyo ngayon, kaya nga usually we talk of syndromes when we're talking of viral infections, kasi marami sa kanila ang nagpo-cause ng gastroenteritis. So we're not talking of signs and symptoms pagdating sa viral infections, rather we're talking of syndromes. Okay? So Virus na nagpo-cause ulit ng gastroenteritis, this forms another distinct genus. Yung coronaviruses ng animals readily established persistent infections, and it includes yung mouse hepatitis virus and avian infectious bronchitis virus. And more for the more on the RNA viruses, we have our retrovirus. So, ano ang alam nyo na with regards to the retroviruses? Anong virus ang kasama dito when we're talking of the retroviruses? Anyone?
HIV.
Yes, okay. So we have here the HIV. So your human immunodeficiency virus. So ang mga retroviruses natin, we have their spherical envelope viruses, and their genome, you have 2 copies of linear positive sense single-stranded RNAs, wherein you have an illustration of the HIV virus on the lower right of the slide. And the replication, di ba sabi natin kanina, na diyan makikita kung ano yung kaagad na pwede mag-undergo ng replication. So bakit sila tinawag na retroviruses din? Because they have to have the reverse transcriptase. Now, virion assembly occurs by budding on plasma membranes, and leukemia and sarcoma viruses of animals and humans, fomiviruses of primates, and lentiviruses, they're all included under retroviruses. So as mentioned earlier, was one of the group, it is the cause of our AIDS or acquired immunodeficiency syndrome. And the orthomixo, ano si mga orthomixo natin? Ito, pag nakita nyo yung mga hemagglutinin at saka mga neuraminidase activity, this is unique to your orthomixo and also to paramyxoviruses. So replication of orthomixo, our nucleocapsid is assembled in the nucleus. Then of course you have your hemagglutinin and the neuraminidase accumulate in the cytoplasm. The virus matures by budding. So all orthomixovirus are influenza virus, but it should not be confused with our coronavirus. Now, paramyxo, so sabi ko nga kanina, similar to orthomixo, but it is larger. Particles are pleomorphic. The human pathogens under paramyxo, we have their mumps, yung measles virus natin, para influenza, metapneumonia, and your RSV or ang ating respiratory syncytial viruses. And Bunia viruses, they're also pleomorphic. And it contains 3 circular helically symmetric nucleocapsids. Replication is in the cytoplasm. Envelope is from the Golgi. So majority of these viruses are transmitted to vertebrates by arthropods except our hantavirus. Kasi ang kanyang mode of transmission is from aerosol o excreta from rodents. Okay, so yung infected rodents. Now, the cause, it's also the cause of hemorrhagic fevers and nephropathy and severe pulmonary syndrome. So, Bornavirus, these are envelope, spherical, 80 to 125 nanometers, and Virus disease, the Borna disease virus is neurotropic. So when we say neurotropic, what do we mean by neurotropic? Nag-ibig sabihin pag sinabi natin neurotropic, saan parte sila maka cause ng infection?
Nervous system.
They're able to get into the nervous system. So it's postulated to have association with neuropsychiatric disorders of humans. So that's your Borna viruses. And then of course, your rhabdoviruses, alam nyo na ito when we say it's the only one. Pag nakita nyo yung bullet form na ganyan, resembling a bullet, then that is your rhabdovirus. And they're measuring about 75 to 180 nanometers. Envelope is 10 nanometers. And the genome is linear, single-stranded, non-segmented, negative sense RNA. Ano sabi natin pag negative sense RNA? Paano ang kanyang replication? Wala siyang wala siyang polymerase. Paano ang replication ng ating rhabdovirus? Anong kapareho niya? Mainit. Anong kapareho ni rhabdovirus kung siya ay negative sense RNA? Okay. Anyone? Diyan pa kayo? Natulog na yata kayo. Hi.
Hello, ma'am.
Gising pa kayo?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Ay, yung mga yes ninyo, parang yes, ma. Kailangan yata muna tumayo, mag-exercise, magtalon-talon muna tayo. Okay? So anyway, I know it's very boring kasi sabi ko nga gusto ko na nga tanggalin kanina ito. But it's in the PowerPoint anyway. And I'm continuing the PowerPoint courtesy of Sir Raymel. So dito sa rhabdovirus, ito yung classic example natin, yung rabies virus. Okay. So I think this is the last among the RNA viruses. We have the Philo Viruses. They're envelope pleomorphic viruses that may appear very long and thread-like. They typically are 80 nanometers wide and about 1,000 nanometers long. Envelope contains large peplomers. Ano na ulit yung ating peplomer? Nandun kasama sa inyong terminology nung lecture ni Sir Tom. Saan nyo makikita yung peplomers na yan? Kasama ba siya ng ano? When we're talking of the peplomers, you have the capsules and what is in the structure of your virus. Okay? So ito yung glycoprotein spikes, which are the subunits of the viral envelope. Yan yung ating peplomers. So, ang kasama sa filoviruses ninyo, you have your Marburg and the Ebola viruses, okay, which causes hemorrhagic fever. Now, when these are the viruses that you need to handle in the laboratory. Remember, we mentioned about a laboratory safety, which will be, I think, next in the slide after the other infectious particles. Then, kailangan dito ng maximum containment conditions. And when we talk of containment conditions, ito yung meron i-consider yung biosafety level ng laboratory and also the biosafety cabinet wherein manipulation of the virus will be done. Okay, so pwedeng BSL-4 safety level, biosafety level ng laboratory, and ang kanyang biosafety cabinet would be your BSC class 3. Okay, so other infectious particles, so you have already your viruses, meron din tayong viroids. Ano itong viroids? So these are small infectious agents that cause diseases of plants. Okay? So, yung mga viroids kasi sabi nga dun sa, all of these, just in case, all of these notes in the PowerPoint were lifted from, ang reference book dito ni Sir Raymel was on Jowetz. Okay? Medical Microbiology by Jowetz. So, These are nucleic acid molecules without a protein coat. Yan ang ating viroids. So there are plant viroids are single-stranded. They're covalently closed circular RNA that does not encode any protein products. Kaya nga hindi sila papasok dun sa definition ng ating classic viruses. None have been demonstrated to exist in animals or humans. Now, another infectious agent or infectious particle is your prions. So you have come across this already noon pa sa microbiology ninyo. So that ano naman ang ating prions? These are infectious particles that are composed solely of protein with no detectable nucleic acid. So kabaligtaran na naman ngayon ni viroids. Sabi natin kanina, si viroids, anong meron siya? Siya ay nucleic acid molecule pero wala siyang protein coat. Si prions naman, meron siyang meron siyang protein coat pero wala siyang nucleic acid. So ito yung yung disease ng prions is called your transmissible spongiform encephalitis. Encephalopathies. Na tinatawag ito pag sa sheep yung TSE na yan yung tinatawag natin scrapie. Pag siya ay sa cattle, yan yung ating mad cow disease. At pag yan ay sa humans, yan yung tinatawag natin na Kuru or yung Creutzfeldt-Jakob disease. I know you have encountered this also in your AUOBF. Okay, so we're done now with the different, infectious particles from the DNA and the RNA viruses to the viroids and the prions. Now we go into the structure, the virus structure. Remember dun sa lecture ni Sir Tom ninyo that di ba, dependent sa virus structure din or the replication of the virus is also dependent on the structure of the virus, even their classification. So that yung virus structure nila, we have here different types of symmetry of these virus particles. So ano ba yung mga techniques na ginagamit to resolve fine differences in the basic morphology of viruses? Remember, sabi natin, we cannot look into the virus structure using now the bright field microscope. Yung presence ng virus into a specimens using the bright field microscope would more be into looking at the cytopathic effects that they produce on the infected cells. But when we're talking of the basic morphology of viruses, anong ginagamit natin techniques? We're using the electron microscopy. We have the cryo-electron microscopy and also yung ating X-ray diffraction. So, ano yung sa electron microscopy? So, how do we do that? Kaya nga ang meron lang, di ba, ang mga meron lang electron microscope is research laboratories kasi alam naman natin kung gaano kamahal ang electron microscope. So, with electron microscopy, this requires the use of heavy metal stain. For example, dyan yung ating potassium phosphotangstate to emphasize surface structure. Now, with cryo-electron, from the word cryo, di ba yan yung sa temperature, so that with cryo-electron microscopy, this uses virus samples, quick frozen in vitreous ice. So with this, sa cryo-electron microscopy, kahit merong quick freezing niyan, hindi nasisira yung surface structure or yung mga fine structures ng ating virus. So with cryo-electron microscopy and the quick freezing still preserves the fine structures of the virus. Now with X-ray diffraction, ito din yung tinatawag natin na if you came across it when you read your module or your book, any book naman, nandun makikita nyo pa rin yan. X-ray diffraction or anong ibang term ng ating X-ray diffraction? Anyone? Anyone? Any two? Any three? Anong ibang term ng X-ray diffraction? Hi.
X-ray crystallography, ma'am.
Yes, it's crystallography. Okay? So this X-ray diffraction or crystallography, may images later on, can provide atomic resolution information. So the specimen must be crystalline, and this has only been achieved with small non-envelope viruses. So ito yung pinapakita yung crystallographic structure ng ating simple icosahedral virus. Okay. So this slide now shows you the first symmetry or yung shape ng ating viruses. Ano yung tatlo? So ano yung tatlo? Na-mention na ni Sir Tom nyo dun sa umpisa ng kanyang lecture. Ano yung tatlong shape ng ating viruses? No, na-mention na yung una dyan. You have the icosahedral pattern or yung tinatawag natin cubic symmetry. Ano yung isa pa? Ay, dalawa pa. Dalawa pa.
Helical.
Tatlo yan. Ha? You have your icosahedral and then
and complex.
helical and then
Complex.
complex. Okay, very good. So, ano yung sa icosahedral? So, yan yung pattern niya. Kaya pag nabibasa niyo yan, sabi niya, yung kasi pag tinignan mo yung structure, hindi mo talaga siya makikita as icosahedral. Rather, ano nakikita niyo na physical appearance niya? It's more of, like if you see the adenovirus on the right upper corner of the slide, ano yung nakikita yung physical shape ng ating virus. It doesn't look like it's an icosahedral, di ba? Hindi nyo makikita yung mga 2-fold, 3-fold, 5-fold na axis. Hindi nyo makikita yung kanyang 20 faces. Hindi nyo rin makikita yung 12 vertices na description ng ating cubic symmetry. So, ang itsura nila, pag tinignan natin sila sa electron microscope, spherical, para rin lang silang bilog-bilog. Okay? But that is our icosahedral pattern. So, yan yung description nila usually when you talk of a cubic symmetry, yung kanyang sinasabi na meron siyang 20 faces, 12 vertices, and then you have the 2-fold, 3-fold, 5-fold axis of symmetry. Okay. So we get into the helical symmetry. So helical symmetry is, of course, ito naman yung itsura niya. These are protein subunits are bound in a periodic way to the viral nucleic acid, winding it into a helix, kaya helical. The nucleocapsid is coiled inside a lipid-containing envelope. So parang barrel yan. There is a regular periodic interaction between the capsid protein and the nucleic acid. And all animal viruses with helical symmetry contain RNA genomes. So all have flexible nucleocapsid except your rhabdovirus. So ito yung itsura ng ating helical symmetry, yung mini-mention kanina, so that you have there the nucleocapsid. Diba sabi niya, the nucleocapsid is called inside a lipid-containing envelope. So that, sabi natin niya, example would be your rhabdovirus. Makita niyo ulit yung rhabdovirus natin dyan, which has the distinctive or characteristic bullet shape. Now, another one that has a helical symmetry is also the influenza virus. Yung TNV, that's your tobacco mosaic virus. Okay? And the third symmetry or shape is the complex structure. Now, from the word pa lang, complex, sabi niya. Pag complex, ano naman yung sa complex natin? So, meron tayong mga viruses na hindi nag-exhibit ng yung unang symmetry or yung ating shape na cubic, or hindi rin siya nag-exhibit ng helical. Hindi siya icosahedral, hindi siya helical. So, mas complicated pa ang kanilang structure. Dito, ang example natin sa mga viruses with this complex structure ay ang ating pox virus. So, tignan nyo yung kanyang brick shape. appearance with ridges on the external surface and a core and lateral bodies inside. So, reviewing lang, you had your icosahedral. Anong itsura nila physically?
Spherical.
Spherical. So that the other one, you have your helical. Anong itsura nila? Para lang silang
Barrel.
barrel shape. Okay, and then you have your complex structure. So that example dito, meron kayo ng Pax viruses. So that is to the symmetry of the dun sa ating virus structure. Now, aside from that, meron din tayong measurement of virus sizes. Kahit gaano pa sila kaliliit. Masabi nga natin, kaya nakikita nyo yung comparison usually. So that dito sa sabi nga natin, di ba maalala nyo yung, I don't know if I included it in the module, yung sa filterable viruses, the search for the elusive virus, na sabi ganun, ba, na filter nila they tried to filter out the virus, pero nung ma-filter na yung specimen, supposedly to have to get the virus or isolate the virus, pag-check nila ng filtrate, infectious pa rin yung filtrate. So, anong ibig sabihin nun? Hindi na filter out yung virus sa gawa ng kanyang sobrang liit. So, ang nagstay lang doon sa filter ay yung bacteria. Although with the discovery of bigger viruses now like your Pandora virus, hindi na rin ngayon lumulusot doon sa filtration yung Pandora virus. Okay? So, yung filterable na property kasi yan, that was unique to viruses. Kasi nga sobrang liliit nila. So, yung direct observation natin ulit sa electron microscope, ito ang pinaka-ginagamit na method for the measurement ng ating virus size. So, yung viruses natin, paano natin sila ma-visualize sa using your electron microscope? Paano natin sila ma-visualize? So they can be visualized from tissue extracts and also in ultra-thin sections of infected cells. So may standard ka dyan. Kung sa bacteriology, meron tayong micrometer that is attached to the, dun sa, ang tawag nyo dun, yung sa pagsilipan natin sa microscope. Meron naka-attach dun na parang na micrometer na magme-measure ng bacteria. But that is of course for the bacteria kasi madali yung i-measure using your bright field microscope. Now it's different also for the electron microscope. But meron din silang standard wherein you can check now for the exact size of the virus. Now, another method that can be used is, of course, we have our sedimentation in the ultracentrifuge. So dito, yung relationship between the size and the shape of a particle and its rate of sedimentation permits determination of particle size. So kasi yung ultracentrifugation, depende yun sa revolutions per minute, di ba? So that para lang ma-pull down ba ang right term? Para lang bumaba yung virus particle. Kasi titignan mo yung rate of sedimentation as to the number of revolutions that you need to take. So doon mo ma-measure yung particle size ng virus. Now, we have here comparative measurements. So pag titignan ninyo itong comparative measurements, si Staphylococcus, of course, when you're trying to check for their sizes, is 1,000 nanometers. Ang ating bacteriophage is 10 to 100 nanometers. Ang ating viruses, there are 20 to 300 nanometers. And there are illustrated here so that, sabi ko nga earlier, if you get into the internet trying to check the sizes, comparing your viruses to other pathogens like our bacteria, makikita nyo yung kung gaano siya talaga kaliit. Although with the discovery nga ni Pandora virus, medyo hindi meron na tayong malaki na virus, hindi na lang puro maliliit. Okay, so this is just an illustration of the sizes of the human DNA viruses and our also RNA viruses, comparing it to E. coli, and to the chlamydia, the bacteria. Okay. So, when we're talking of the virus structure, ito yung illustration natin. This just, kita nyo na yan, di ba? Sabi natin, virus structure, simplest is you have your nucleic acid plus the capsid, which makes your nucleocapsid. Now, aside from your nucleic acid, which could be DNA or your RNA, meron din tayong structural proteins and enzymes na yan ngayon will make up your nucleocapsid. So, di ba, pag sinati mo yan, si nucleic acid at si capsid, yun ang protect dun sa iyong nucleic acid. Yun yung pinakasimple na structure ng ating virus. And that is what you call your naked capsid virus. Kasi wala siyang envelope. Now, yung nucleocapsid mo, dinagdagan mo ng glycoproteins and membrane. Ano ba itong glycoproteins and membranes mo? Yan yung composition ng envelope. Yan ngayon yung tinatawag natin na envelope virus. So, ano naman ngayon so we're done with the symmetry and then also with the size. Now, ano naman yung chemical composition ng ating viruses? When we're talking of the chemical composition. So we have, of course, you have the viral protein, you have your nucleic acid or the viral nucleic acid, meron tayo ng viral lipid envelope, and meron din tayo ng viral glycoproteins. So going into the viral protein, ang structural protein ng viruses, marami silang function. So what are the different functions? So you can see there, ano yung major function ng ating, the major function or the major purpose of these viral proteins. First is, kita natin yung first dyan sa ating slide, it facilitate transfer of the viral nucleic acid from one host cell to another. Okay? And viral proteins also serve to protect the viral genome against inactivation by nucleases. And also, they participate in the attachment of the virus particle to a susceptible host, susceptible cell. And they also provide structural symmetry of the virus particle. Now, the virus, the enzymes that are maybe present in very small amounts, they also determine the antigenic characteristic of the virus. So these are essential for the initiation of the viral replicative cycle. So yan, later on, makikita nyo pag we get into the individual viruses, you'll get to see the viral proteins that are present and their functions. Now, for the viral nucleic acid, ano yung viral nucleic acid, di ba? Sabi ni Sir Tom, it can either be just an RNA or a DNA. It can never be both. So that this encodes the RNA or DNA that encodes the genetic information necessary for replication of the virus. So the genome may be single or double-stranded, as you could have, sandali lang. Ate. Sorry. Ate. Diyan si Ma'am Meng may dinadaan. Okay, so the genome may be single or double-stranded, as you could have, na-notice nyo kanina nung pinipresent natin or tinitignan natin yung mga RNA viruses. Pwede silang circular or linear and segmented or non-segmented. So, yung type ng nucleic na acid niya, yung kanyang strandedness, ano yung strandedness natin, di ba? We say single or double-stranded. And yung size, ito ang major characteristic na ginagamit for classifying viruses into families. So, parang kunyari, tignan nyo dyan, yung size ng ating pox virus is about 375 kilobase pairs, whereas sa ating picorna, you have your 4 kb at ang coronavirus is also 32 kb. Okay? And next is your viral lipid envelope. So, with the lipid envelope, so it is, di ba makita nyo yung illustration on the right kung paano nakukuha ng mga viruses ang kanilang envelope. So that these lipid envelopes, they are acquired when the viral nucleocapsid buds through a cellular membrane in the course of maturation. So that yung budding process, it depends, di ba sabi natin earlier din, depende yan, yung replication nila, so that the budding process depends on the replication strategy of the virus and the structure of the nucleocapsid. So, nakita nyo yan, di ba, pag bud out niya, may masasama na part nung host cell dun sa bagong virus particle. So specific phospholipid composition of a virion envelope is determined by the specific type of cell membrane involved in the budding process. So lipid-containing viruses are sensitive to treatment with ether and other organic solvents. So what happens? Kasi bakit nagiging sensitive sila? So nagkakaroon ng disruption or loss ng lipid. So pag nawala yung lipid envelope na yun, na-disrupt or nasira ba? Tama ba yung term? Nasira is that nawawalan din ng infectivity yung virus. Okay? So non-lipid containing viruses are resistant to ether. Kaya nga yung ating coronavirus or SARS-CoV-2, di ba sabi natin, they are sensitive to even just the simple alcohol or to some detergents. And of course, the viral glycoproteins, ano man yung viral glycoproteins natin, di ba sabi natin kanina, yung sa lipid envelope is from the host cell. Now, these viral glycoproteins, they are virus encoded. So, this surface glycoprotein, they attach the virus particle to a target cell through they attach the virus particle to a target cell by interacting with a cellular receptor. So this surface glycoprotein, di ba sabi natin, if you remember about the specificity and tropism ng mga viruses, depende yan sa receptor na dapat present dun sa target cell. So that these glycoproteins are also important viral antigens and interaction of particle with neutralizing antibodies. So X-ray crystallography again provide insight into the antigenic structure and functional activities of viral glycoproteins. So going back, ano na ulit yung chemical composition ng ating viruses? We start with, ano yung ating
Viral protein.
Okay. Viral proteins. Then you have your
Viral nucleic acid.
Nucleic acid.
the viral nucleic acid. And then
Lipid envelope.
Viral lipids.
you have your
Viral lipids.
Lipid envelopes.
lipid envelope. And then
Glycoproteins po.
you have your glycoproteins. So next, we get into the cultivation of viruses. Now, remember that, di ba sinabi natin, you're all aware already of how difficult, kasi hindi sila kapareho ni bacteria natin na kumuha ka lang ng sample, nilagay mo sa media. It could be a general medium, or you have the differential, selective, and you have the enriched. It's different for our viruses. Kasi ano ba sabi natin? When you define what a virus is, you always say they are, yung simple, pinaka-simple ng definition ng viruses. Ano? simplest definition of viruses. They are asan na kayo?
Smallest infectious agents, ma'am.
Simplest description. Bakit hindi natin sila mag-grow sa culture media? Bakit kailangan natin ng mga cell lines?
They are minute.
Ha? They are?
Not living cells.
Small
They
ma'am.
are obligate intracellular.
Sa living cells lang sila nagre-replicate.
Yes. Kailangan nila ng living cell. Diba yan ang isang point of discussion. Noon sila ba ay considered living or not? Because hindi naman sila pwedeng mabuhay pag wala silang living cells. Kaya tawag din silang parasites. So that as you have said, they're obligate intracellular parasites. So that to cultivate the cultivation of those viruses, kailangan natin ng cell culture. So dati din ginagamit din yung kita nyo yung dati noon parang nasabi natin sa microbiology, yung inoculation sa embryo, kasama din yun sa fertile eggs. So kailangan natin dito ng mga cell lines. And we have here the 3 basic types of culture when we're trying to cultivate our viruses or try to grow these viruses. You have the primary culture, the secondary culture, and the continuous cell line. So that pag sinabi nating primary culture, this is made by dispersing cells from freshly removed host tissues. Ang problema with primary culture is that they're unable to grow for more than few passages in culture. So we're not talking of subculturing kasi pag viruses ang pinag-uusapan. Ang term na ginagamit is more on passages sa culture. Okay? So sa secondary culture naman, ano ginagamit dito? Using diploid cell lines. Ano na ibig sabihin pag diploid? Diba meron kayong haploid, meron kayong diploid. Anong ibig sabihin pag diploid? Meron siyang diploid.
Shared chromosome po.
Chromosomes.
2 copies of chromosome.
Okay, yun yung ating mga diploid cell lines. And these have undergone changes that allowed their limited culture but retained their normal chromosome pattern. Now, sa mga continuous cell lines natin, these cell lines, continuous cell lines, are culture capable of more prolonged growth that have been derived from, sila nang galing, diploid cells or from malignant tissue. Kaya makita yung mga Vero cell lines, HeLa, yung mga ganun na cell lines na ginagamit para sa cultivation ng ating viruses. Now, yung type ng cell culture na gagamitin for viral cultivation, again, of course, we said earlier, depends on the sensitivity of the cell to a particular virus. So ano na namang characteristic ng virus yung kinoconsider dyan when you're trying to grow them on different cell lines? Talking of anong characteristic ng virus? Sabi natin, depends on the sensitivity of the cell to a particular virus. That is what we call specificity. Okay, wala kahit hindi sila basta-basta pwedeng tumubo sa kahit anong cell line lang. Dapat yung cell line na ginagamit natin to cultivate these viruses, meron sila nung receptors. Okay. So, detection of virus-infected cells so that multiplication of virus can be monitored by. Paano natin malalaman ngayon kung present ba siya? Doon sa kumuha ka ng sample, nilagay mo siya sa iyong cell line. So paano mo makikita ngayon kung meron ba yung virus, present ba, at nagre-replicate ba. So that there are different methods or modes of how you can see whether the virus is replicating or not, kung present siya or hindi, is that in development of cytopathic effects. So that when we talk of cytopathic effects, di ba, you came across this one na din. Siyempre, may histology na kayo, nag-histopath na rin, may histopath na rin kayo. Ano yung mga cytopathic effects? Ano yung mga cytopathic or cytopathologic effects?
Structural changes po, ma'am.
So what happens to the cell? Yes, there's a structural changes. So that what happens? Ano ma-observe ninyo? Di ba pwedeng parang sa...
Lysis po, ma'am.
Ha?
Lysis po, ma'am.
Okay. So you will see cell lysis or necrosis. And then, pwede kayo makakita ng...
Inclusion bodies.
Yes.
Vacuoles, ma'am.
Then, yes. Ano pa? Meron kayong giant cell formation, meron kayong cytoplasmic vacuolization. So lahat yan nagpapakita ng cytopathic effect. Remember that when a virus is able to attach and penetrate the cell, ano ang gagawin niya dun sa cell? Di ba? It will lead into either the death of the cell or gagawa siya ulit dyan ng mga inclusion bodies niya or the rest that are mentioned in the slide. So another one would be by appearance of a virus encoded protein. Like for example, ano yung mga virus encoded protein? Sabi natin kanina, ano yung mga virus encoded protein natin? Ito yung sa ating glycoprotein. So we have here your example would be the heme agglutinin ng ating influenza viruses. Then another would be detection of virus-specific nucleic acid. Saan natin nakikita ito? Yung sa mga virus-specific nucleic acid. This would be by which molecular techniques? Molecular techniques would be by polymerase chain reaction or your PCR. Another one would be the adsorption of erythrocytes to infected cells, which we call the heme adsorption. And of course, E is the viral growth in an embryonated chick egg may result in, of course, the death of the embryo. Like for example, in encephalitis viruses, you have the production of pox or plaques or plaques. And yung plaques kasi yan yung na-form pag ganyan meron na-infect yung cell ng virus. So this can be seen with herpes, smallpox, and vaccinia. And again, for the development of heme agglutinins, which happens with influenza viruses. So this is how the different CPE would look like, although hindi siya masyadong ma-appreciate dito, but you can get into the internet. This was lifted up from Jawets. So that pag enterovirus yung nilagay natin dun sa cell line, so there will be, ano ang CPE na makikita natin? There will be rapid rounding of cells progressing to complete cell destruction. Sa herpes viruses, yung sa B, you will see focal areas of swollen or rounded cells. So you should be able to differentiate kasi yung normal cell sa cell na nagbibigay ng meron ng cytopathic effect due to the presence of the virus. Then sa paramyxovirus naman, you can see focal areas of fused cell, which you call now your syncytium or your syncytia. And heme absorption, ano sabi natin kanina sa heme absorption? It is the absorption of this RBC to infected cells. So the erythrocytes adhere to those cells in the monolayer that are infected by a virus that causes a hemagglutinin to be incorporated into the plasma membrane. Yung monolayer, di ba lagi natin nababanggit yan, lalo sa virus isolation. Kasi pag grow yung cells na lalagyan, sa natin i-cultivate si virus, kailangan yan isang, kaya mono, di ba we say mono is just for one. You have to see a monolayer of those cells where you will try to grow or where you would try to ang tawag doon sa term na yun para mag-replicate or mag-multiply yung virus. Okay? So, for the inclusion bodies, sabi na natin kanina, another one would be to the formation of inclusion bodies so that these inclusion bodies, dyan yung sa maraming viral infections, yung inclusion bodies ang site ng development ng ating virions. Remember, when we're talking of virions, these are already your complete virion particle or yung infectious particle or yung tinatawag natin na viral factories. Okay? Kasi di ba yung mga virions yan yung marami ng dumaming particles while they are inside the cell. Kaya tinawag siyang viral factories. So, inclusion body formation are produced during the course of viral multiplication within dun sa cells. Now, they become larger than the individual virus particle and often have an affinity for acid dyes. Ano yung example natin ng acid dyes? So we have here may histopath na kayo, di ba? Ano yung ating mga acid dyes? Example would be your eosine. Now, they may be situated in the nucleus like for the herpes virus, sa cytoplasm kung sa pox virus, or in both sa measles virus. So, the presence of inclusion bodies may be of considerable diagnostic aid. So, pathognomonic sign na yan. Sa nakita mo si Negri body sa specimen, then that is already diagnostic for rabies or for the rabies virus. Okay. So, ano ba yung mga example ng inclusion bodies natin? So, you have the Negri bodies for your rabies virus. You have the Torres concealment bodies for yellow fever. And then the multinucleated giant cell, yung pinaka-common na intracellular inclusions that are seen on the Zang smear for our herpes virus. So ito examples ng mga inclusion bodies. Then, aside from that, now you can also quantitate. So na-culture na natin sila, nakita natin yung kanilang CPE, just so we know which virus may be present. So we can also quantify for the viruses. Kanina, di ba, nagtingin na tayo ng kanilang symmetry, nagtingin tayo ng size. Now, we can also quantify or what we call quantitation of viruses, wherein we have the napindot ko. Bago lang siya nag-mute, parang nakita ko biglang nag-mute. Okay? So, we have quantitation of viruses. Meron tayong two methods. You can do the physical method and the biological method. So, sa physical method, we have quantitative nucleic acid-based assays, which we have mentioned earlier again, which is your PCR. Ano magagawa ni PCR? This can determine the number of viral genome copies in a sample. Copies, di ba alam natin sa polymerase chain reaction, what are we doing here? We're amplifying the nucleic acid. So, makikita ito both sa both infectious and non-infectious are detected. So, yung sa hemagglutination assays or what we shortcut natin as HA. So, yung mga viruses na may hemagglutinine, ano ulit ang ginagawa ni hemagglutinine? Na meron ito sa ating influenza viruses. Anong nangyayari pag yung ating virus may hemagglutinine? Lamesero na kayo. What happens when the virus has a hemagglutinin? Anong pwede niyang gawin? It can. Hi. Hello. What happens?
Clumping po, ma'am,
Clumping, yes,
of
or
red cells.
yes, the clumping or the agglutination of your RBC or your red cell. So that these are easy and rapid method of quantitating this type of viruses. So those that have, of course, the hemagglutinin. So we have again electron microscopy. So pwede na naman natin bilangin ang ating virus particles directly sa ating electron microscopy. This is with comparison to standard suspensions ng latex particles na magkapareho or similar sa size. Okay, so may standard ka. Bibilangin mo na naman yung mga virus particles. Now, for the biologic, bakit tumigil? Okay, for our biologic method, so we have here the endpoint biologic assays, which depends on animal death, animal infection, or cytopathic effects in tissue culture at a series of dilutions of the virus being tested. So, the titer is expressed as ID50. Ano yung ID50 natin? That is your infectious dose at 50. So, it is the reciprocal of the dilution of virus that produces the effect in 50% of the cells or animals inoculated. So that is your infectious dose 50 or ID50. For the plaque assay, di ba na-mention naman natin kanina, the plaque or the formation of pox, this is widely used assay for infectious viruses. So what do you see when you have the formation of plaque? These are multiple areas of infection formed after, multiple cycles of replication and cell killing. So that example here, you have the herpes and vaccinia, wherein they form pox onto chorioallantoic membrane of embryonated eggs. So such viruses, this herpes and vaccinia, they can be quantified by relating the number of pox counted to the viral dilution inoculated. Okay, so ayan yung pag-measure natin. Those are the biologic methods when we would like to quantify or in the quantitation of viruses. So, purification of virus particles. So you had this, if you still remember, this was discussed in your molecular biology. Ba, nung second year kayo? So that purification, for purification studies, the specimen, these are usually large volumes of tissue culture medium, body fluids, or infected cells. So, ano yung unang gagawin ninyo when you're trying to purify? Kasi kailangan mo when you're doing the test, so you will need pure viruses. Para rin lang sa bacteriology when you would like to isolate a pure colony para wala kang contaminated, walang contamination. So that the first step in purification is the concentration of the virus particles, by which method you have, of course, precipitation and heme agglutination. So that with precipitation, you do this with ammonium sulfate, ethanol, or polyethylene glycol, or by ultrafiltration. Remember, bakit ultrafiltration? Di ba kanina nga sabi natin, kaya dun sa article ng The Search for the Elusive Virus, wherein pinadaan sila sa through filtration, pero ano tignan yung filtrate, infectious pa rin, which means that hindi na-keep yung virus dun sa filter. Now, for hemagglutination and elution, this is more to use sa concentration ng orthomixovirus. viruses. So, yung mga orthomix natin or the other viruses, pag yan na-concentrate na, di ba sabi natin, the first step is the concentration, then pwede na natin i-separate from the host material by, anong gagamitin natin? Again, by differential centrifugation. You had this already. You have density gradient centrifugation or column chromatography and also electrophoresis. Now, yung ating mga icosahedral viruses, mas madali silang i-purify kesa sa mga envelope viruses. And remember and take note that it is difficult to achieve complete purity of viruses. Then, how do we now? So we have had concentrated, and then we are able to separate. So now, how will we be able to identify? Paano ba na natin masasabi na yung particle na yan ay virus talaga? So yung na-identify, yung bang na-concentrate natin, na-separate natin. So ano yung mga criteria bago natin masabi na yung particle na yan ay siya yung ating hinahanap na o tinatry na i-identify na virus. So we have different criteria, and we have there that first is that the particle can be obtained only from infected cells or tissues. Okay. Second is that the particle from various sources are identical regardless of the cellular origin. Number 3, particles contain nucleic acid and sequence that is not the same as the host cell. And the degree of infectivity, the degree of infective activity of the preparation varies directly with the number of particles present. And another one would be the destruction of the physical particle is associated with a loss of viral activity. Remember yung kanina sabi natin, if it is sensitive to ether, sa mawawala dun sa lipid envelope niya, masisira yung lipid envelope, rendering the virus now hindi na siya infective. Okay? So, number 6, certain properties and infectivity must be shown to be identical. So, ano yung mga properties na yun? Pwede yung kanilang sedimentation behavior, nakita natin kanina sa quantitation ng viruses, or pwede rin yung kanilang, ano pa ba ang pwede nilang property? Yung kanilang pH. pH is stability of the virus. Now, we can also identify a particle as a virus if when an anti-serum prepared against the virus should react with the characteristic particle or vice versa. Again, we go back into the specific antigen antibody reaction, diba? So that yung anti-serum prepared against the virus should react with the characteristic particle, or baliktaran, yung antigen naman ang prepared, hindi yung antibody. Now, another one is a particle should be able to induce the characteristic disease in vivo. But this one is only if it is possible or feasible to do. Okay, yung criteria na yan. But otherwise, marami naman tayong criteria na pwede nang pag-basean para lang masabi natin na yung particle na yun ay virus particle. And the last, of course, but not the least, the passage of the particles in tissue culture should result in the production of progeny with biologic and antigenic properties of the virus. Okay. So, yan yung mga criteria natin. So, after, siyempre, nag-cultivate tayo, you try to grow, you try to also quantify. Now, you can also now try to identify kung yan ba ay virus or not. Okay, so, going into the identification, we still have to consider, again, laboratory safety. So alam naman natin like ngayon, di ba? Ang dami nating question when we see medical technologies or molecular biology technologies getting infected with the SARS-CoV-2. Ang tanong lagi, bakit ganun? Bakit sila nahawa? Di ba naka-PPE naman sila? What happened? So that we are aware that karamihan kasi ng mga viruses that are human pathogens, syempre they can cause what we call laboratory acquired infections. So, natawag natin laboratory acquired kasi nagawa ang ayan during the manipulation of the virus in our effort to try to identify para lang maging magawa ng diagnosis. Now, so with this, we know that these viruses, they are potentially hazardous if proper technique is not followed. So, ano yung mga technique na yun? From the donning and the doffing of the PPE, from the collection of the specimen to the processing of the specimen, hanggang sa ito ay ma-dispose or sa kanyang disposal ng ating specimen or ng mga tawag natin doon yung mga hindi lang yung specimen after you have processed the specimen. So the common hazards that might expose laboratory personnel to the risk infections are, of course, we have here aerosol. Ano yung aerosol? Yung naging point of discussion din natin, point of discussion with regards to the coronavirus, whether it is being aerosolized or it is sa droplet lang. So that when we know that aerosols are generated by, paano ba magkakaroon ng aerosolization? These are generated by homogenization of infected tissues, centrifugation through ultrasonic vibration or broken glass rays. Kaya meron na tayo ngayon ang mga centrifuge na ginagamit, especially when sa mga molecular laboratory, meron na silang cup para maiwasan yung aerosolization ng ating sample. Now, another one would be ingestion. Although wala nang mouth pipetting ngayon kasi marami na tayong mga micropipettes or automatic pipettes, meron pa rin siguro yung mga resource limited na laboratory so that ang ingestion is made possible when you're doing mouth pipetting. Now, another one would be eating or smoking in the laboratory. Contaminated yung pagkain mo. So, nakain mo na rin si virus. Or, meron yung virus sa kamay mo so that hindi tayo naghuhugas, kulang ng hand washing, so that pwede mo rin ma-ingest si virus. Another one would be skin penetration. This would be through needle pricks, meron kang broken glassware, hand contamination by leaking containers. So if you go back to your handling and processing of specimens, di ba? Tinitignan nyo muna kung walang leakage. And handling of infected tissue or animal bites. And pwede rin yan sa splashes sa ating eyes. Kaya meron tayong the appropriate PPE. You try to get to see ano ba ang pwede na maibibigay na hazard no ating specimen during the manipulation or during the processing of the specimen. So those are the common hazards. Through aerosols, through ingestion, through skin penetration, or splashes in the eye. Now, good biosafety practices. So what are the good biosafety processes? Mula nung senior high kayo hanggang ngayon, mamaya, you will need this also in your internship. The irony of it, irony of it is that I have been sent an invitation from Bacolod nga, San Agustin something. I just read the email this morning. And I feel guilty that I haven't gone into the joint memorandum circular. We have a JMC for your internship. Kasama daw doon sa letter kasi kasama daw doon sa JMC ang biosafety training for incoming interns. So it's good that we have it here now. Although we'll have maybe a bridging program with regards to that. Sorry, I still have to get into the joint memorandum circular with regards to internship. Hindi ko pa siya nabasa. I attended the webinar on this, kaso wala nga naman walang na-finalize during the webinar. So good biosafety practices, what are these? So we need additional precautions and special containment facilities when working with high-risk agents. Kaya alam naman natin kung ano ng high risk, di ba? You remember the risk grouping of these pathogens. You have to get into ABSA kasi the risk grouping of the pathogen will tell you of the appropriate good biosafety practices that should be in place. So training in and use of aseptic techniques. Again, siyempre, hindi naman tayo nag-laboratory ng face-to-face. You were not able to perform all of this. The proper manipulation or yung paghawak man lang ng ating Petri dishes or paano ba yung aseptic techniques because of course, the aseptic techniques where you're inside the biosafety cabinet or nasa bench top lang ba tayo. The interdiction of mouth pipetting. Interdiction means elimination. Sabi ko nga kanina, hindi naman na ginagamit ito dahil we already have automatic pipettes, micropipettes, wherein you were able to do it nun sa mall bio experiment, yung kaisa-isang experiment na nagawa sa mall bio. And of course, again, it's always reiterated. Kuminsan kasi medyo pasaway pati mga staff. We admit to that. Sabi natin, no eating, drinking, or smoking in the laboratory. And the use of the PPE, not only the use, but of course, what is important here would be the proper donning and doffing of the PPE and the proper or the use of appropriate, hindi naman OA na paggamit ng PPE. Yun talagang kailangan lang natin when we are doing or trying to do the processing of the specimens. And also, the sterilization of experimental waste. Sabi ko nga kanina that we need to, the manipulation starts from specimen collection, processing hanggang sa disposal. And when we're talking of sterilization, ano yung pinaka-common method and the most used method just to come up with the sterility assurance level bago natin i-dispose ang ating mga experimental waste. Ano yung pinaka-common method of sterilization?
Incineration po, ma'am.
Alin-alin? Again?
Incineration po.
Bawal incineration, darling. Meron tayong Clean Air Act. We do,
Autoclave.
bawal sa Philippines. Yes, it's the autoclave. Okay? When we're talking of sterilization, although common sterilization, pag doon tayo sa aseptic techniques, is flame heating of inoculating loops. But when we're talking of experimental waste, like for example, you have here yung ginamit natin ng mga cell lines. Siyempre, may tumubo na doon ng mga viruses. So we do sterilization, which is through the autoclave. Alam naman natin, pag yan na autoclave and the autoclave is functioning properly, then we're sure that lahat ng microorganisms or viruses or pathogens that may be present ay napapatay. I don't know if the right term is napapatay. So we get back into your sterility assurance level. And of course, the use of biosafety hoods or what we also call as our biosafety cabinets. Again, I know I have lectured, I hope I did lecture ito, yung nandito ako kung sa inyo o sa medicine, that the biosafety level of laboratory is not synonymous to the class of the biosafety cabinet. Okay, hindi pag sinabing BSL2 kami, so ang iisipin mo dun, meron kang biosafety cabinet class 2. Hindi sila synonymous. So when we're talking of biosafety hoods or our biosafety cabinet, babalik na naman tayo sa ating risk assessment para matignan mo anong risk group ba ng iyong pathogen na mina-manipulate. Then, doon mo masasabi kung anong dapat na class ng biosafety ang gagamitin natin. But normally, sa mga hospitals, sa microbiology department, syempre yan, we have the biosafety cabinet or biosafety hood class 2. Okay? Kasi yung mga class 3, ito na yung mga ginagamit sa mga research laboratories. And of course, another biosafety practice, a good biosafety practice would be immunization. If relevant vaccines are available. This is the reason why we got a survey amongst all the sections, amongst all sorry, BS, MLS, incoming interns. Although hindi porket nag-survey kami, sorry ha, it's not for anything else, but the survey doesn't really tell that everybody will be going for internship. Sabi nga ni Dean, ma'am, sabihin mo ha na yung survey is only for allocation of vaccines. It's not because sinurvey natin sila, automatic interns na sila. So, relay ko lang. But I know with your efforts, honest efforts, I hope you all get to be eligible for internship. Kahit 576 hours lang ang required na i-render ninyo sa hospital. But that will be after deliberation, meetings, and everything, still with your parents and also with the university and affiliation centers. So yan yung mga good biosafety practices that you have to keep, anong tawag doon? Isa puso, ganun sa Tagalog. Dapat isa puso ninyo. Hindi pwedeng mawawalan kayo ng focus pag nasa lab na kayo, especially in this time of pandemic. Hindi natin alam yung inyong mga specimens, although limited, hindi kayo lalabas, hindi kayo mag-flebo. It's just more of trying to enhance yung mga skill-based dapat na competencies na meron kayo bago kayo graduate. These biosafety practices are very important. So we'll get into this like the use of biosafety cabinets, ano ba yung mga meron sa laboratory later on siguro. Yun nga sabi ko kanina, we may try to organize parang bridging program lang, yung mga ini-expect na dapat alam nyo pasok ninyo for your okay? So, now we get into reactions muna ng ating viruses to physical and chemical agents. So, para rin lang itong dun sa decontamination, sterilization natin dun sa bacteriology. So, that first to consider here is physical. Ano yung physical? Diba? That is exposure to heat and cold. But that we say here that icosahedral viruses, they tend to be stable, losing little infectivity after several hours at 37 degrees centigrade. Yung ating mga envelope viruses, there are more heat labile. Di ba sabi natin na basta yan envelope, mas madali silang i-destroy. So that them being heat labile, so they lose infectivity after prolonged storage even at minus 90 degrees centigrade, and they are sensitive to repeated freezing and thawing. So yung viral infectivity nitong mga envelope viruses are or is destroyed by heating at 50 to 60 degrees centigrade for 30 minutes. Now, exception dito is your hepatitis B virus and C. polyoma virus. Now, some viruses are preserved in the dry state. Ito yung tinatawag nating lyophilization at 4 degrees centigrade or even at room temperature. So, yung another one would be stabilization of viruses by salts. So, ano yung mga ano yung ginagagawa ng salts as to making our viruses stable. Diba? Pag sabi natin stable, hindi mo yan siya mababago yung kanyang infectivity or yung viability ng virus. So that many viruses can be stabilized by salts in concentrations of 1 mole per liter. So that example dito is that, sabi nga natin earlier dun sa unang slide, di ba? Dun sa ating effect ng heat and cold, that yung viruses are not inactivated kahit na i-heat mo siya at 50 degrees for 1 hour. Di ba sabi natin kanina, heating at 50 to 60 degrees at 30 minutes? Heating to 50 or 60 degrees for 50 minutes, masisira yung mawawala ang infectivity ng virus. But once these viruses are stabilized nga by salt, sa concentration lang na 1 mole per liter, hindi mo na din sila ma-inactivate kahit i-heat mo sila even for longer period. Remember dun sa ating decontamination, sterilization noon na yung contact time ng heating and the temperature will be dependent on the bacteria. So ganun din dito. Once the viruses are stabilized, yan nga, with the addition of salt, sa concentration lang na 1 mole per liter, hindi mo na sila ma-inactivate. Kahit i-heat mo sila at 50 degrees centigrade for even a longer period of time, parang 1 hour. So, ano yung mga salts na ginagamit dyan? Like you have your magnesium chloride, you have your magnesium sulfate, and sodium sulfate. So this magnesium chloride, this stabilizes the pig coronaviruses and reoviruses. Yung mag sulfate stabilizes your orthomyxo and paramyxoviruses, and sodium sulfate, tama ba ako? Na2SO4, is that your sodium sulfate stabilizes your herpes viruses. Ano ang importance or ang advantage ng stabilization nitong mga viruses by using all of these salt solutions? Saan siya magagamit, maa-apply? This can be applied sa preparation ng vaccines. Remember, naging issue din yan sa pagpili natin. Anong vaccine ba ang gagamitin natin with the SARS-CoV-2? So, kinonsider pa rin yung kanyang cold chain, yung mas feasible na gawin para ma-maintain yung stability ng vaccine. Remember, the vaccine has either the inactivated virus or part of the virus, diba? Yung sa mga lectures, merong mRNA or you have the proteins. So, providing stability to this, the stability of viruses, importante yan sa preparation ng vaccine. So that yung pagdagdag, ba tawag dyan, paglagay or whatever, ng ating salts for the stabilization of the virus. So, ma-maintain mo yung potency for weeks. Mapapatagal mo yung potency niya for weeks at even just ambient temperature. So, yan ang importance ng stabilization ng viruses gamit ng mga salt solutions. Ang concentration lang ay 1 mole per liter. Now, how about para nabubulol na. Ang hirap ng lecture pala ng tuloy-tuloy. How about for the pH naman? So that when we're talking of the pH, they are stable between pH values of 5 and 9. And all viruses are destroyed by alkaline conditions. Now, variation in pH influence heme agglutination reactions. Now, ang ating mga enteroviruses, they are resistant to acidic conditions. Now, for radiation, alam natin na, di ba, usually yung sa radiation, yung infectivity is most sensitive. Infectivity is the most radiosensitive property because replication requires expression of the entire genetic contents. So that irradiated particles that are unable to replicate may still be able to express some functions in the host cells. So when we're talking of radiation, ano yung mga ginagamit natin? Pwede yung ultraviolet, pwedeng X-ray, pwedeng high energy particles inactivate viruses. So next one is we have ether susceptibility. So this has been mentioned a lot kanina dun sa ating different RNA viruses, whether they are ether sensitive or they are ether susceptible. So it is used to distinguish, sabi nga natin kanina, envelope viruses from non-envelope viruses. We also have detergents. So what are these? They can be non-ionic or the anionic detergents. An example of your non-ionic detergents, we have our non-idet P40 and Triton X100, commercial yung mga yan. Anong function niya? They solubilize the lipid constituents of viral membranes. Ang ating anionic detergents, you have your SDS, your sodium dodecyl sulfate. Ano ang ginagawa niya? Sinusolubilize niya ang viral envelopes so that it disrupts the capsid into separated polypeptides. How about for your formaldehyde? So these are just now your chemical agents that are used for the inactivation of viruses. So that formaldehyde destroys viral infectivity by reacting with the nucleic acid. So viruses with single-stranded genomes are inactivated much more readily, and formaldehyde has minimal adverse effect on the antigenicity of proteins, and therefore this has been used frequently in the production of inactivated viral vaccines. For photodynamic inactivation naman, we know that viruses are penetrable to a varying degree by vital dyes. Ano yung mga vital dyes na yan? We have our toluidine blue, meron neutral red, and also your proflavin. So that yung mga dyes na yan, what do? They bind to the nucleic acid, and the virus then becomes susceptible to inactivation by visible light. So neutral red is the one that's commonly used to stain sa plaque assays. And antibiotics and other antimicrobial, parang nangangalahati pa lang tayo. Sana tayo. Antibiotics and other antibacterial agents. So what are these antibacterial agents naman when we're talking of inactivation? We have antibacterial, antibiotic, and sulfonamides together with the quaternary ammonium compounds, organic iodine compounds, and alcohol are ineffective. So we need sa larger concentrations of chlorine are required to destroy viruses than to kill bacteria, especially in the presence of extraneous proteins. So for example, an example natin pag nasabi natin larger concentrations, like for example, you have a stool sample. Doon sa stool na yun, pwedeng i-inactivate ng chlorine ang alin? Anong pwede meron? Sa stool sample, pwedeng i-inactivate ng chlorine ang typhoid bacillus. Okay? But pero yung chlorine na kaya niyang i-inactivate si typhoid bacillus, hindi niya ma-destroy si polio virus doon sa doon sa stool sample kung present siya. So parang in short, kung doon sa stool sample mo meron kang typhoid bacilli and then you have the polio virus, si typhoid bacilli mai-inactivate ni chlorine. Kailangan mo ngayon ng larger concentration para naman matanggal mo yung polio virus kung present siya dun sa feces. Now, meron tayong, di ba, we have the alcohols. Ano yung mga alcohol na ginagamit natin ngayon? Recommended, we have your isopropanol and ethanol. They're also relatively ineffective against certain viruses, especially ang ating mga picorna virus. Now, for the common methods of inactivating viruses for various purposes, ano yung mga methods? Yan is, alam natin that we need to inactivate viruses for various reasons. For example, you need to sterilize laboratory supplies and equipment. Kailangan natin i-disinfect ang surfaces. I will not use disinfect surfaces for skin, rather antiseptic for skin. We need to make drinking water safe and produce inactivated virus vaccines. So, meron tayong iba-ibang methods dyan And meron din tayo mga chemicals that are used for these purposes. So you have sterilization, you have surface disinfectants, we have skin antiseptics, and then sa vaccine production, na-mention natin kanina, diba, pwede natin gamitin yung ating formaldehyde, and then, bakit may message? Formaldehyde, we also have beta-propiolactone, soralen, plus ultraviolet irradiation or detergents. Now, for ito, I know that you are already aware of the viral replication cycle. You have had it, di ba, sa cytogenetics ninyo, meron na doon yung viral replication. Na-touch din sa mol bio. So, review na lang ito. Or kayo naman mag-explain. Pagod na si ma'am ng 1 hour lang. 1 hour. What are the different steps in viral replication? Anyone from the group? Alam nyo na ito. Wala na yung 19. Kanina 189 kayo. Ngayon 170 na lang kayo. O, nag-171. 170. Ha?
Sige, ma.
Okay, go. Steps in replication.
Ito yung last month.
Ha? Brief lang. Brief, description of the different steps of replication. Hi, anyone? Imagine niyo lang yung parang sa viral pathogenesis na rin. Ano ang unang step dyan? Paano mangyayari yung unang step dyan? Anong kailangan sa attachment? Na-mention na natin kanina sa iba-ibang natin. Topics discussed earlier. What do you need when you talking of the attachment of the virus sa kanyang specific cell na pwede lang niyang i-infect? Hi, anyone from the group? Eunice, are you there? Tawagin ko yung mga... Yes, Eunice. Sabi ko pa naman, tatawagin ko yung mga classmates. Eunice. Ano yung steps natin sa replication? Ayan na, ano mo na lang. First
Attachment po, penetration or engulfment, tapos uncoating po, ma'am.
Ayan yung first part niya. So, anong kailangan mo sa attachment?
Yung virion po, ma'am, mag-i-interact po siya dun sa cell surface po. Meron po
Ano
siyang
dapat meron sa cell surface? Ha?
Meron po siyang specific receptor site po.
Kaya dapat may receptor si cell kasi kahit pa hindi siya kaya nga at lagi niyong tatandaan yung specificity o tinatawag nating tropism pag sa viruses. Kasi kailangan yung cell na yun meron siyang receptor, meron tagatanggap, may tatanggap doon sa virus. So that here, sabi natin dyan, ano yung mga receptor sites natin? So you have their different receptor. Ang ibig sabihin yan, parang for example, yung ating Picornaviruses, saan siya magbabind lang sa protein sequences. And sa orthomyxo, ano naman ang kanyang receptor? That will be oligosaccharides. Now, common knowledge ito that, di ba, pinakamagandang example dito, when we're talking of attachment, doon sa unang step sa viral replication would be your HIV, your Human Immunodeficiency Syndrome, wherein saan siya nag-a-attach is it will bind to the, ano nakalagay dyan, to the CD4 receptor cells ng ating immune system. So that, yung iba naman, dyan nilagay lahat dyan, yung ating rhinovirus, magbabind siya sa ICAM1, And then si Epstein-Barr virus ay marirecognize niya yung CD21 receptors sa B cells. So lahat yan, mas alam nyo na yung mga yan kasi may immunosero kayo. And immunosero lang ba? Tama, immunosero lang yung mga yan. Okay, so you need the receptor. Kaya nga yan lagi, paulit-ulit sabihin ni ma'am, yung specificity and your tissue tropism. So pag naka-attach na siya, ano ang next? Parang kunyari may friend ka, parang ganun. Naka-attach na siya dun sa cell surface. So that the next would be
Penetration.
or engulfment. Kaya parang may bucket dun, papasok na siya, makakapasok na siya. So that the virus particles taken up inside the cell, this is accomplished by receptor-mediated endocytosis, pwedeng direct penetration across the plasma membrane, or pwedeng fusion of the envelope with the plasma membrane. So after binding, mapapasok na siya, so yan, tatawag natin na penetration or engulfment, nasa loob na siya ng cell, so that uncoating now takes place. So doon sa uncoating, andyan yung physical separation of the viral nucleic acid. So that is either your DNA or your RNA from the outer structural components of the virion so that it can function. Then the genome may be released as free as in picorna virus or as a nucleocapsid as in the case of our reovirus. Now, the nucleocapsid usually, anong meron ang ating nucleocapsid? They usually contain polymerases. Okay? So, nakalagay ulit yan sa inyong slide. The RNA virus usually release the genome into the cytoplasm, whereas the DNA virus most release their genome into the host nucleus. So, that is the first part of your replication. Mag-attach siya. Naka-attach, mag-penetrate, naka-penetrate, mag-uncoat. Re-release na niya ang kanyang nucleic acid. Diyan na magkakaroon ngayon ng the expression of viral genomes and synthesis of viral components. So the mRNA now are transcribed from viral nucleic acid for successful expression and duplication of genetic information. So babalik na naman tayo kay negative at saka kay positive strand. So that yung mga negative strand natin na RNA, kailangan nilang isupply yung sarili nilang RNA polymerase para ma-synthesize ang mRNA. Now, for the morphogenesis, the last part of the replication, you have your morphogenesis and release. So newly synthesized viral genomes and capsid polypeptides assembled together to form now your progeny viruses. Whereas icosahedral capsids can condense in the absence of nucleic acid, nucleocapsids of viruses with helical symmetry cannot form without viral RNA. Non-enveloped viruses accumulate in infected cells and the cells eventually lyse and release now your virus particles or yung ating virion or yung ating infectious particle and tuloy ulit ang ating replication cycle. Okay? So ito yung diagrams lang, example ng viral growth cycles. Yung sa left natin, this is the growth cycle of non-enveloped double-stranded. So, kita nyo yung viral DNA niya, kaya double-stranded DNA virus. In contrast, doon sa inyong right, andyan yung growth cycle ng positive sense. So, pwede siyang magtuloy-tuloy ng replication. Diba sabi natin kasi meron silang mRNA. So, ayan yung growth cycle of a positive sense, single, so makita nyo yung RNA niya, single-stranded RNA. So, ayan yung mga processes niya from the attachment to the penetration to the uncoating and then sa next expression, and then you have your morphogenesis and then hanggang sa release. Okay? So, we move on into, so that is your replication, viral replication or multiplication. Review lang naman na yan sa inyo. So we move on to the natural history and modes of transmission of our viruses, of the viruses. So pag sinabi natin ecology, diba? Ano yung ecology? The study of the interaction between living organisms and their environment. So that the mode of transmission utilized by a given virus depends again on the nature of interaction between the virus and the host. So that viruses may be transmitted in the following way. So meron tayong direct transmission from person to person by contact, which is the major means of transmission. It includes droplet or aerosol infection. Pwedeng, dito ang examples natin, we have the influenza, measles, and smallpox by sexual contact in the case of papillomavirus, hepatitis B, herpes simplex type 2, and the human immunodeficiency virus, or the hand-to-mouth, hand-to-eye, or mouth-to-mouth contact with herpes simplex, and exchange of contaminated blood in hepatitis B, and again with HIV. So, indirect transmission by the fecal oral route, ito yung sa ating enteroviruses, or by fomites sa ating Norwalk and Rhino. Animal to animal with humans as an accidental host, ito yung sa ating rabies. And then transmission by means of an arthropod vector. So, saan ito? Ito yung sa ating, when we're talking of transmission by means of an arthropod vector, this is with alin? Our type of viruses. May exam yata kayo, mukhang nagre-review kayo, baka may quiz kayo sa next na ano.
Arbor Viruses, ma'am.
Ha? We have your Arbor Viruses. Okay. So, moving on, ito yung parang cycle. Ang pinapakita lang yung different transmission patterns recognized among arthropod-borne viruses. Pwedeng human arthropod cycle. So, that dito sa human arthropod cycle, so it is a commonly, saan ito nakikita? Yung human arthropod cycle, pinaka-common na arthropod-borne infection sa kinagat ni lamok si tao. Kailangan kagatin ulit ng arthropod si tao para ma-complete yung cycle. Saan? Anong viral infection? Arthropod.
Dengue, ma'am.
Dengue, thank you. And another one is the lower vertebrate arthropod cycle with tangential infections on humans. Dito na cycle, pag tinignan mo yun, pagkatapos nung arthropod to a lower vertebrate arthropod pumunta sa human, tangential, the human here is a dead end host. So ito yung cycle na more common transmission mechanism. An example dito would be with our St. Louis encephalitis. And yung isa pa is the arthropod cycle with occasional infection of humans and lower vertebrates. Example natin dito yung sa ating lacrosse encephalitis. Dito sa cycle na to, yung virus, pwede nakita nyo arthropod to arthropod, so that yung adult arthropod, pwede niyang i-transmit yung viral infection or yung virus sa kanyang offspring, saan through egg or yung tinatawag natin na, ano na yung pag vertical, pag sa human. So sa arthropod, mapupunta dun sa kanyang offspring through the egg. And then the cycle, pwede niya magpatuloy with or without intervention of a viremic vertebrate host. Kaya yan lang yung mga, it's nice to know lang naman. So what are the different factors that are contributing to disease emergence. When we're talking of disease emergence, bigla na lang meron ulit na nawala na, bumalik siya. So bakit merong bagong disease? Like for example, the SARS-CoV-2 that we have now. So alam natin na the spectrum, alam natin na yung tawag ba doon? Ano ba tawag doon? Yung ayaw kong gamitin yung word na emergence ulit. Yung pagkakaroon ng mga infectious diseases na dati wala are nag-expand. Expand ba yun? Dumadami yung cases natin. Maraming bagong agent. You have novel viruses or novel bacteria. At saka yung mga sakit na dati nating alam na under control, nagkakaroon ulit ng incidence. Okay? So that pati yung pag-evolve ng pathogens, di ba sabi natin, you learned in your host-microbe interaction na in as much as we are trying also ways to eliminate or to neutralize sa viruses, yung virus is naghahanap din ng ways itong mga viruses just to be able to cause the infection. Lalo ng viruses, they need to get into a cell para matuloy yung mamadali si ma'am. Tuloy-tuloy yung buhay nila kasi kailangan nga, di ba? Kaya tinatawag natin sila na obligate intracellular parasites. So, ang ating mga viral diseases, they emerge following one of the three general patterns. Ano yung mga yun? Siyempre, magre-recognize ka muna ng bagong agent. Merong abrupt increase sa illnesses caused by an endemic agent. Diba? Remember, when we talk of endemic agents, the presence of that vector in a certain location lang. And then, yung invasion ng new host population. Ano yung new host population? Parang kunya hindi naman nakaka-infect ng humans. Pero gawa ng certain alterations, yung mga host microbe interaction or the host virus interaction, naging host na rin ang tao. Okay? So, what are the different factors now that contribute to this disease emergence? You have environmental changes. You look around you. Okay, so we are thankful that Bising did not have a landfall. So kasi di ba summer na summer, meron tayong super typhoon. So these are due also, this emergence of diseases are also due to environmental changes. Meron tayong deforestation, meron tayong changes sa ating ecosystem. Ano yung mga changes kung hindi baha, tagtuyo, parang mga ganun. So those are all contributing to disease emergence. Human behavior. What happened to us? Ano na yung mga behavior ng tao? Nagko-contribute din yan sa emergence. Then you have socio-economic and demographic phenomena. Like for example, poverty, ano pa ba? Population growth, pwede migration. Travel commerce, kakanta tayo na it's a small world after all. Why? Kasi you can go anywhere and hindi naman anytime, lalo na ngayon. Madali na lang mag-travel so that we have made the world smaller, sabi nga nila. So that one disease that may be endemic in one place can now get into another place. You carry the disease with you. We also have food production. So ano naman sa food production? Anong kinalalaman ng food production into the emergence of diseases? So, nababago kasi you have to cater to the needs of the population. Kailangan mo mag-mass produce or kailangan mo din ng changes in the methods of food processing and sa packaging. So, this can also contribute. And then the healthcare, what happened to healthcare? When we're talking of healthcare, meron ng, ano maisip nyo sa healthcare? Health. Helpline. Ano pwede sa healthcare natin na mag-contribute into disease emergence? Transplantation, organ transplantation, blood transfusion. Sa microbiology, antimicrobial resistance, gawa ng inadvertent use of antibiotics. Then we also have microbial adaptation. Again, babalik na naman tayo sa host microbe. So naka-adapt na rin itong mga microbes, nakatingin na rin sila ng ways on how to live dun sa kanilang host. Then of course, meron na naman dyan yung ating development of antimicrobial resistance, and public health measures. So ano ba nakikita nyo ngayon dito sa emergence ng ating SARS-CoV-2 at bakit tayo nagkaroon ulit ng surge? Ano ang nangyari sa ating mga public health measures? What do you think that is contributing now again to the surge of the SARS-CoV-2? Akala natin wala na, tapos biglang ayan na naman siya. What happened with our public health measures? Nagkaroon ba nag-relax ba tayo? Nagkaroon ba
Yes, ma'am.
nagkaroon ba ng breach sa prevention measures? Nagkulang ba tayo sa mga trained personnel? Sa communication? And all of those, lahat yung public health measures. Okay? So, pwede rin tayo mag-isip-isip ngayon, are we contributing now to disease emergence? Okay? So, what's next? What's next? After you have learned almost everything ba? Or after learn about the viruses, kasi nasa virology na tayo ngayon. And it is very timely kasi meron tayong pandemya na ang may kagagawan ay virus. So after all of those, we have all of those things that we learned about the viruses. Ano kaya ang susunod? We have a novel virus. Now we have climate change. You have environmental changes. And all of this, ano ang kasunod? What is your role as public health workers later on? Alam mo, dun sa huling webinar as regards to public health, wherein Dr. Belisario was mentioning about the gaps in public health na remember kasi sa UP Manila, ang tawag nila sa medtech, di ba? Hindi sila BSMLS, sila ay Bachelor of Science in Public Health. Pero may dalawa kasing track yan, pwedeng epidemiology, pwedeng MLS, na ganun. So that with this, after learning about, the information about viruses, and of course, sama nyo na rin yung mga huling na aral ninyo about bacteriology, what's next? What is your role as public health practitioners later on? Either, as a health authority, or you will be a medical laboratory scientist, or you'll be proceeding to your medical courses. So what could you contribute? I-stop ko muna to para mag-ano muna tayo ng video ka. Makita ko naman kayo. Start video. Paano mag-stop na ulit? Skip? Ay, stop sharing pa lang. Stop share. Okay. So what's next? Ano ito? Or anyone from the group. What's next? Close natin yung chat box. Asan na kayo? Anyone?
Prevention of disease, ma'am.
O paano anong role ninyo sa prevention? O dito muna tayo sa what can you do at this moment in our life? Parang ganun. Ngayon na nandito tayo sa pandemya. Ating SARS-CoV-2 pandemic. What can you do as a public health worker or as a medical laboratory scientist? Meron bang natulong yung naaral nyo ngayong araw na ito? Meron ba kayong natutunan na bago? O meron bang na-correct na mga misconceptions? Ano? Wala. May klase pa kayo? Titignan ko lang. Bakit walang sumasagot? Juna V, where are you? Juna V? Wala din? Okay, so...
Hi, ma'am.
Hi, Anje.
Hi.
Hi, Zarina.
I think we
Yes.
could help battle misinformation po, ma'am.
Okay, how can you help dun sa misinformation? Specifically?
Ano po kasi, as hard as it could be to believe, marami pa rin po kasi hindi naniniwala sa COVID.
Okay. So marami pa rin hindi naniniwala. So parang binabaliwala pa rin nila si COVID. So with this, di ba, ang daming lumalabas sa social media, what you can do if there are misinformations or yung mga lumalabas sa social media, you can always try to correct din. So you start among yourselves. Kahit naman, syempre maraming pilosopo pa rin kasi nasabi, oh, wala yan, ganyan lang. Fake news yan, hindi totoo yan, ganyan, ganyan. So you can start from, alam mo yung sa circle ninyo muna, and then you come up with, magkakaroon kasi ng domino effect yan. Lang, para na lang yung sa paggamit ng mask, pag lumabas ka. Mga iba ayaw pa rin mag-mask. Wala naman daw kausap. Malayo naman. Yung mga ganun, so that pwede kayong mag-convince, tapos syempre gawin yung evidence-based, ganun nga sa science, dapat evidence-based. So tignan nyo na lang, sabi nyo, tignan nyo yung mga nurses, yung mga frontliners. Bakit nagka-COVID pa din sila? Kung hindi totoo na ganyan kaano ang COVID. So something like that. So I tried to come up with that. What's next? Kasi nga, parang kung minsan, knowledge is there. Alam mo, natututo or hindi natututo. Pero kahit pa paano, may nakikita ka, may naririnig ka, may nababasa ka. But then the attitude and the practice that goes with the knowledge, yun tayo nagkakaproblema. Yun ang problema natin ngayon. So as MLS of public health workers nga, I hope you did learn something from today, no matter how boring it is in viruses. Lalo na kaninang enumerate natin yung mga RNA viruses. I know how boring it could be, but you just have to be enthusiastic enough din para to learn of the different viruses. Okay, so as I have mentioned earlier, yung next topic kasi natin is viral pathogenesis. So I'll just send it to you. I can pwede mag-record or I can give you the notes, whichever you would like. Kung ano mas gusto nyo, kung na-recorded ba or just the plain notes, so you can study it on your own, and then any things that needs to be clarified. So we can discuss on that next meeting, pero we'll start already with the individual DNA viruses. We start with the DNA viruses. Binigyan na kayo ng mga mnemonics ni Sir Tom ninyo. So that if you master, pag alam nyo na si DNA viruses, di sa kasama nun, lahat ng ibang viruses, RNA na yan. Alamin nyo na lang whether this is a positive sense or a negative sense. Although, mas importante ang diagnosis. And I will not require you naman to memorize, ako single-stranded siya, double-stranded siya, positive sense siya, negative sense siya. What is important is you know what a positive sense RNA is and what a negative sense RNA is. Yung mga talagang kailangan na malaman nyo as medical laboratory scientists. So that is for this afternoon session. You'll have your post-quiz on Tuesday. There will be no pre-quiz. Instead, I will just mag-review na lang muna kayo for the post-quiz. Medyo mahaba ang introduction. So the pre-quiz will be considered as a recitation during the discussion na lang. Okay, so keep safe everyone. Thank you for your afternoon. Thank you for listening. Sabi ko nga, I hope you did get something. You did pick up something from today's lecture. So stay safe everyone. And cluster 1, mag-prepare na kayo. Dapat champion tayo. Joke lang. Enjoy lang natin yung college days natin, kahit virtual. We need also to relax and enjoy. Kahit alam ko nag-academic relief kayo, madami pa rin kayong inaral. So relax din. Paminsan-minsan. Huwag tuloy-tuloy na relax ha. Paminsan-minsan lang. Okay, so good afternoon everyone.
Thank you,
Bye.
ma'am.
Thank you, 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 ma'am.